Would you like to get your product on the market faster? Would you like to receive shorter letters from the FDA? If so, you are on the right track by attending this workshop. Good morning. My name is Kelley Burridge. My talk today will focus on common deficiencies from a quality perspective. The goal here is to help you avoid the mistakes of others and know what to submit to work towards those first cycle approvals. First, I'm going to present several reminders for items to include in the various modules of an ANDA application. The focus of my talk is going to be on 3 different types of Q3 studies. And lastly, I'm going to give you some overall tips and two resource slides that have a wealth of information. Again, these are my points of view and my tips to help you get that first cycle approval. This slide highlights items that are commonly missing from different modules of an ANDA application. There are a large number of FDA literature articles on this topic. They are penned by FDA authors, and they are listed on resource slide 1. Some items just to remind you, provide analytical method protocols for all of the tests in your drug substance and your drug product specifications. Provide us with the analytical method validation or verification information as appropriate. Include transfer reports if the facility that's going to be performing the routine testing is not the same as the lab that developed the method. Remember to include force degradation data with mass balances. If you analyze the same sample using your assay and your related substances methods, and you can demonstrate that you achieve a mass balance, that's pretty strong evidence that your methods are stability indicating. Provide a detailed description of your drug product. You could say white cream. That's not as helpful as saying that it's, there's no phase separation, it's homogeneous, it's free of foreign matter. Include a description of your packaging. Also include a test for packaging container interaction and whether your labeling maintains its integrity. On stability, samples are generally stored in horizontal and vertical configurations. Remember to let us know, is it horizontal with the cap up or is it horizontal with the cap down? That may seem like a small detail, but again, all that we have to go on is the information that you provide in your submission. So if you don't mention somewhere, we don't know. As Priyanka mentioned, you want to check for a product-specific guidance. If you have a critical functional excipient in your formulation, such as a preservative or a penetration enhancer, remember to include an assay for that component in your specifications, justify your range, and provide us with the analytical method validation information. Next, I'm going to focus on these Q3 studies. So the first one is a Rheology study. Shown here are loading and unloading curves. These also go by the name of shear stress versus shear rate curves, or you may have heard them referred to as up and down curves. These curves really provide a wealth of information about what type of fluid your drug product is. So the 3 things I want you to remember from this slide in terms of characterizing your product, is it Newtonian or non-Newtonian? Does it have a yield stress? And is it time dependent or independent? For Newtonian fluids, the shear stress varies linearly with the shear rate. That's the case for a lot of topical products that are solutions that don't have any kind of component in the formulation that modifies the rheology. However, most of the topical products, the ointments, the gels, the creams, they have items in the formulation that are going to affect the rheology. In a lot of cases, those are non-Newtonian. And what is meant by that is simply that the shear stress does not vary linearly with shear rate. Many topical products are shear thinning, so the viscosity, again, the viscosity is the slope of this curve, so that the value that you're going to get depends on your shear rate. Now, when you're thinking of shear stress and shear rate, one way to think about this is, has anyone ever made ice cream using an old-fashioned ice cream maker? Right? You have a cylinder, you put your ingredients in, you have a mixer. The shear rate is related to how fast the mixer is moving and the fluid inside. The shear stress is how much force has to be exerted to get that mixer to continue to move. We generally ask for people to provide loading and unloading curves over attainable shear rates until a low or high plateau is identified. It's helpful if you provide your product's data and the RLD on the same plot. Another thing to look for is whether or not the loading and unloading curves fall on the same line. If they do, then your fluid is not time dependent. However, if you load and then you unload and the curves don't overlap, then there's some time dependence of your fluid and you have a little bit more of a complex situation. So you need to be more careful when you develop your method for measuring viscosity. You may have to do a little bit more method development there, but again, these things sometimes are as simple as allowing your sample to rest for 10 minutes before you take a measurement. The most common attribute, the rheological attribute in drug product specifications, is the viscosity. And here, again, the topical products span a wide range of viscosities. So anything from a solution that pours easily to an ointment that doesn't move when you invert the container. I often see someone proposing a specification of 1,000 to 1,000,000 centilpares. And that would be equivalent to saying that your product has a viscosity somewhere between honey and peanut butter. I have a feeling patients are probably going to notice if your product is varying that much. So again, you want to provide us with comparative data to the RLD. You want to justify your specification range. Use the exact same method to measure both samples. If you use different methods, we can't compare the data. Try to reduce variability so that the measurements that you're taking will reveal any real trends if they're there. Typically, we see ranges on about an order of magnitude are generally what is observed for this type of product. If your product contains a solid API or you have an emulsion, we're probably going to ask you for API particle and globule size studies. Again, this is a whole subject in and of its own, but there are some common techniques to do these measurements. Laser light scattering is one. Another common one is microscopy. So shown on this slide, we have pictures of solid APIs and also different types of emulsions. A lot of times I'll get a data table and someone says, these are my results, these are my particle sizes, and look, it's very repeatable, but I don't know how you've measured it, and I don't know what measure you're reporting. So provide us with information. If you're using an automated technique to measure particle size with microscopy, make sure that you let us know that your software is actually correctly identifying those particles. So an example of that is provided here in the middle. 2 hands here. So this is an example of the raw data image showing the particles. And then on the right, this is what the software detects as the particles. So those are now outlined. So if you show us this, then we have more confidence that your software is correctly identifying particles and providing a size distribution that makes sense. So again, provide comparative data to the RLD. Provide us with comparative photomicrographs of the same magnification. You may want to use different magnifications to highlight different attributes of your products. And remember to define your measure and also provide us information about your technique. Remember to label your figures. Tell us what it is. Provide us with the sample information. Identify the age of the sample at the time of the testing. For the RLD, how close it is to expiry. Where possible, try for age-match comparisons between your product and the RLD, especially for attributes that vary with time. Provide us with complete test reports, especially when asked. This helps us to assess the method and know that your metrics are meaningful. Explain what your data mean. You are the expert. Justify the ranges that you choose for specifications and processing conditions. Again, by providing us with comparative data to the RLD and product development data. If trends are apparent on stability, discuss them and what they mean for drug product quality. Here are the literature articles that list the common deficiencies in ANDA applications. At the bottom is a link to a video of another quality presentation on the same topic. Here I've listed a few relevant USP standards. The 2 ISO standards are actually cited in the USP, and those are really great resources for doing particle size analysis using microscopy. And of course, our website has a wealth of information, including many guidance documents. Usha will now present the OGD perspective.
Are you ready for more tips for faster approval? Here it comes. Today I'm glad to be here to present common deficiencies cited by the bioequivalence section in ANDAs for topical products. These are my views. Presentation focuses primarily on in vitro release test and deficiencies related to in vitro permeation test for these topical products. Presentation is divided into 2 topics, IVRT, starting with general information on IVRT, pivotal study comparing drug release results between test and RLD should be performed as specified in the USP General Chapter 1724. As you all aware, detailed description what should be submitted for IVRT and IVPT is described detailed in PSG of product specific guidance of acyclovir-cream. So what should be submitted for evaluation of IVRT? IVRT method development report, method validation report. Here is, I want to get your attention that you should submit 2 separate validation reports, one for IVRT method and one for analytical method validation. IVRT pivotal study report and SOP related to all these reports. In IVRT method development, information should be provided to support the selection of method parameters, membrane, and selection of receptor solution. For example, here the selection of the membrane should be supported by giving good linearity and precision of the drug release rate. IVRT method, which is used in the pivotal study, should be validated. And due to time limit, I'm not going to details of these validation compounds. The IVRT method selection and discrimination ability is mainly supported by 3 concepts: sensitivity, selectivity, and specificity, as shown here. And you are recommended to submit the data in both raw data and as a graphical representation. More method validation components are shown here. Turning our attention to the most important common deficiency observed for IVRT study includes missing experimental details like what is the speed used for stirrer, and when do you stop during sample collection? Is membrane soaked or not soaked? If it is soaked, how long? Degassing of the receptor medium, and sample withdrawal, the accurate amount of sample withdrawal for each cell, amount of sample loaded per each cell. I would like to recommend please submit experiment details not only in the pivotal study but in all the reports during the developmental of IVRT studies and the validation reports. Coming to the data analysis, not just reporting the data analysis, it is recommended to provide the detailed description how the data is analyzed. For example, if a membrane selection, for a membrane selection, it is recommended to submit stepwise procedure how you are calculating the binding percentage assay values. Most of the deficiencies observed are missing raw numerical data containing information about dilution factor and quality, including quality control samples in IVRT sample analysis. IVRT data, raw data should be submitted in the form of SANS transport format. It is recommended to submit both reports for analytical and IVRT method, and also SOPs for both analytical and IVRT study. Coming to the next topic, IVPT. IVPT is the comparing cutaneous PK of a drug comparing test and RLD using excised human skin mounted on a qualified diffusion cell system. As I explained before, in IVRT, these are the reports should be submitted similar to IVRT. However, in addition, in IVPT study, pilot study report also should be submitted. IVPT method development should include information for the selection of method parameters. As shown here in the graph, product dose amount is selected based on the maximum flux. Experimental conditions and apparatus should be qualified and appropriately validated, and these are the following validation components recommended to submit in method validation report. IVPT pilot study is recommended to conduct to estimate number of donors required for the pivotal study. It is recommended to use multiple skin donors to accurately and appropriately estimate number of donors required for the pivotal study. If you don't use multiple skin donors in pilot study, it may not give correct estimate for the pivotal study. Parallel assessment should be performed with a third product, and results should not be combined with the IVPT pivotal study. Coming to the pivotal study design, non-dosed control skin section from each donor should be included. Pre-dose zero sample from each diffusion cell should be included. Duration of an IVPT study should specify maximum flux and a decline in the flux. Coming to the important and final part of my presentation, deficiencies for IVPT studies. These are the common deficiencies we observed in our survey. Not including control skin section, insufficient sampling times. Again, as I explained before, skin from only single donor is included in the pilot study. Skin donor inclusion exclusion criteria not submitted. Randomization sequence not submitted. No rationale for clinical irrelevant dose. Details about experiments are missing, like about temperature, size of the tissue mounted on the transdiffusion cell, skin conditions, either occluded or unoccluded, speed of stir bar, dosing and sampling design. For statistical analysis, final data set should include balanced number of replicate skin sections for each treatment group across all donors, and missing raw numerical data with information like dilution factor. Again, I emphasize that Please include quality control samples during your IVRT and IVPT sample analysis. Missing IVPT data in SANS transport form. Incomplete study reports for both analytical and IVPT. For example, if you change any procedure during your pivotal study, you should validate that procedure and should submit method validation. For example, if you use a membrane for a skin under the skin, you should able to provide sufficient justification and validation of that method. In summary, IVRT, IVPT data is reviewed by Division of Bioequivalence. Method validation should be performed using validated sample analytical procedures. Inadequate submission of the data may often lead to the delay in the review process and the final approval of the application. I would like to thank you, each and every person on this slide, for giving me this opportunity. Thank you.